Okay, here we are in the second lecture of endocrine. Today we're going to start looking at some of the actual glands and specific hormones of the endocrine system, and that will be the trend over the next several lectures. I have this chapter broken into 6 lectures where we will look at a specific gland in each of the lectures. Now that we're actually beginning to look at some of the specifics, it's a real good idea for the remaining lectures beginning today to have your lab chart with you as you watch these lectures, because if you haven't already filled it out from your outline, you can definitely fill it out over the coming lectures. So as always, let's begin with our not really attendance questions. Again, these are really going to come from Bio 137 as they tie into today's lecture also. Where are proteins that are destined to leave the cell synthesized? How do those proteins leave the cell? And what is released from the axon terminals at a synapse? The first 2 questions are going to come from the cell chapter in Bio 137. Where are proteins that are destined to leave the cell synthesized? These are going to be synthesized or made on the rough endoplasmic reticulum. Remember the network that we pass through, we start on the rough endoplasmic reticulum. A piece of that RER breaks off, it travels to the Golgi, carrying that synthesized but incomplete protein. The Golgi then takes that protein, modifies it, and finalizes it. And then a piece of that Golgi breaks off. Now we have a vesicle carrying that finished protein. So how do those proteins leave the cell? Well, that vesicle carrying the finished protein travels to the edge of the cell. It merges with the plasma membrane, and then we have exocytosis of that protein. Now, relating that to neurons, let's combine that information with information about synapses and see what is released from the axon terminal at a synapse. Well, it's a neurotransmitter. That's the chemicals that neurons use to communicate with each other or with other organs in the body. But those neurotransmitters typically are proteins, so they follow that path that we just looked at. And that's going to tie into today's lecture and somewhat to all future lectures when we're talking about protein hormones. But specifically, we're going to be looking at neurotransmitter type protein hormones today.
So let's go on and talk about the gland that most of us are familiar with, the pituitary gland. And the pituitary gland, if you've ever taken a biology class or an anatomy class in high school or middle school, you've probably talked about the pituitary gland. And you probably heard it called the master endocrine gland, which the reason they call it that is because it does have control over most of the other endocrine glands. But what we're going to learn today and in the next lecture is that really the pituitary gland has its own master, and that's the hypothalamus. Really, the hypothalamus would be better called the master pituitary gland because it controls the master endocrine gland, and the hypothalamus actually controls the pituitary, and then the pituitary controls the rest of the endocrine glands. But depending on which part of the pituitary gland, there's going to be some really different mechanisms at play. Today, we're going to look at the posterior pituitary gland, which is what's here in kind of this tan or yellow color. And then tomorrow, or the next lecture, is going to look at the anterior pituitary gland. But before we actually start talking about the pituitary gland and its hormones, first I want to talk about the structure of the pituitary gland. Well, the pituitary gland really is 2 separate glands, the posterior and the anterior. And even though it's classified kind of as one gland, it really is 2 completely separate structures. They're made of different material, different types of tissue. and they have completely different origins if we go back and look at the developing embryo. So, first I'm going to go and show you a picture that your book uses as far as development goes. This is on page 631 of your textbook, and this is the developing embryo, and up here this is the developing brain area. Right here is the developing hypothalamus and posterior pituitary. So this is neural tissue. And then also down here in the pharynx, we've got developing salivary glands, and a piece of that salivary gland actually breaks off and starts to go upward. It migrates upward and bumps up against that developing hypothalamus, which is extending down to form the posterior pituitary. And they kind of bump up against each other, and that glandular material eventually becomes the anterior pituitary. The anterior pituitary is glandular tissue. The hypothalamus, which is neural tissue, a part of the brain, extends downward, so this is still neural tissue, and forms the posterior pituitary gland. And the anterior pituitary gland actually kind of bumps up against and wraps around the posterior pituitary gland at this structure right here. So the whole pituitary gland, both the anterior and posterior, hang underneath the hypothalamus from this little stalk. And this stalk is called the infundibulum. The infundibulum. Now we'll come back to that stalk in just a minute. But the pituitary gland and up here the hypothalamus, they're completely surrounded by blood vessels. There's capillaries around the hypothalamus. There are capillaries around the anterior pituitary gland, and there are capillaries around the posterior pituitary gland, which we don't see in this image, but we will in just a moment. So there's lots of capillaries here. And the pituitary gland sits right underneath the hypothalamus, as we said, hanging from this infundibulum. Now, think back to Bio 137 when you were learning the structure of the skull. When you looked inside the brain case, there was that weird little structure called the sella turcica. And the sella turcica was this little cavity, and that cavity is where the pituitary gland sits. So the pituitary gland sits in the sella turcica, in the skull, just beneath the hypothalamus, hanging from the infundibulum. Now, anterior pituitary gland, that glandular tissue, we're going to look at that in our next lecture. The rest of this lecture is going to be focused just on the posterior pituitary gland, which again, remember, is neural tissue, and its relationship to the hypothalamus. Now, not only is this posterior pituitary, not only is it neural tissue, it's specifically axon terminals. So think back to the structure of an axon. You have the cell body with the dendrites, you have a long axon, and then it ends in axon terminals. Well, the cell bodies are up here in the hypothalamus. The axons extend downward through the infundibulum, and the axon terminals are what make up the posterior pituitary, which sometimes is called the neurohypophysis. The neurohypophysis. Now, those axons, we can see here, they're passing through the infundibulum. The glandular tissue that makes up the anterior pituitary gland is kind of wrapping around that infundibulum. And here's that capillary bed that I mentioned that surrounds the posterior pituitary gland that we didn't see. That is actually right here. If we go back to that other image, we can see all of those different blood vessels. Here's around the infundibulum, here it is around the anterior pituitary gland, and here around the posterior pituitary gland. So lots of blood vessels. and there are those axon terminals. Now, when we talk about the posterior pituitary, there are going to be two hormones that are associated with the posterior pituitary gland, and that's what I have over here on the left. It's ADH, surprise, surprise, that's the first hormone that we're going to talk about, and oxytocin, abbreviated OT. Now, most of the hormones that we talk about, we're going to use abbreviations. So make sure to familiarize yourself with those abbreviations. ADH, antidiuretic hormone, also called vasopressin or arginine vasopressin, and OT, oxytocin. Both of these are posterior pituitary hormones, but there's going to be something very important with both of these two hormones, ADH and oxytocin, that is completely different from every other hormone that we will talk about. These hormones are released from one gland, the posterior pituitary, but they were actually made in another gland. They were made in the hypothalamus. So now when you start to see questions, either in lecture or in lab, you have to be very, very careful with how the question is worded. Where is ADH made? It's made in the hypothalamus. Where is ADH stored? It's stored in the posterior pituitary. Where is ADH released from? It's released from the posterior pituitary. So these two hormones, ADH and oxytocin, are made in the hypothalamus, but they are stored in and released from the posterior pituitary. But we're actually going to get a little bit more specific. When we look up here at the hypothalamus, there are actually a few different areas where we find clusters of cell bodies. Now, remember when we talk about clusters of cell bodies in the central nervous system, we gave those special names. We called them nuclei. One cluster of cell bodies is a nucleus. More than one cluster of cell bodies are nuclei. So we have a nucleus here called the periventricular nucleus because it's in one of the ventricles of the brain. It's actually near the third ventricle of the brain. And then we have another one that sits just above the optic chiasma, and this is called the supraoptic nucleus. Now, next class, we're going to see that there's another bundle of these cell bodies, but they're not going to play a role in this. So we're only looking at the periventricular nucleus and the supraoptic nucleus. Now, the periventricular nucleus, these are the cell bodies that make oxytocin. The supraoptic nucleus, these are the cell bodies that make ADH. So think about those not really attendance questions. These neuron cell bodies, like all cells, contain rough endoplasmic reticulum. In the periventricular cell bodies, that rough endoplasmic reticulum is making oxytocin. That oxytocin travels down the axons and it's stored in these axon terminals. If we look at the supraoptic cell bodies, here in the rough endoplasmic reticulum, it's making ADH that then travels down the axons and it's stored here in these axon terminals. Now, every other gland that we talk about and every other hormone that we talk about is going to be made and released from the same gland. The same gland that makes it releases it. But that is not the case for the posterior pituitary. So now, this part, I'm sorry, it's going to seem like I'm reading from you, or reading to you, which really I kind of am. Unfortunately, there's not really a whole lot of chance in this chapter for me to kind of make this a little entertaining, so bear with me. ADH, also called vasopressin. What did we say its stimulus was, or its stimuli were? Well, its primary stimulus should be second nature to you by this point. Increased plasma osmolarity. But it's got some other stimuli also. In addition to increased plasma osmolarity, low blood volume, low blood pressure, but also pain and some drugs will stimulate the release of ADH. It is inhibited by adequate hydration. So when you've got plenty to drink, when you are well hydrated and your plasma osmolarity is low, then we inhibit the release of ADH. Interestingly, though, also alcohol. So for those of you that drink, think about what happens shortly after you start drinking alcohol. You start peeing a lot. You pee a lot more than the actual amount of alcohol that you brought in. And why is that? Because alcohol inhibits ADH. What was the job of ADH? To reabsorb water. Well, if you block ADH, if you inhibit ADH, you're not going to reabsorb as much water, so you will urinate a lot more and your urine volume will increase. That is why you pee so much when you drink alcohol. Now, go a little bit further. If you're peeing a lot, you're losing a lot of water. Your blood volume is lower and you're becoming dehydrated. And the next morning, what do you feel? Hungover. You have a terrible headache. And the reason for that is because you lose so much water, you become so dehydrated, your brain actually starts to shrink. And as it shrinks, it pulls on those meninges, and that's what you're feeling when you have a hangover. Functions, now, something we're going to see at this point, I'm not going to read every single thing in this outline because I've already filled it out for you. Some of this is going to be on you guys to learn. So for function, we've already talked about. We said that the DCT and the CD start to make those aquaporin type 2s, and we've talked about the function for those. So I'm going to skip over that. But I do want to talk about two disorders associated with ADH. Hypersecretion of ADH, when there's too much ADH being released, that causes a disorder called SIADH, or Syndrome of Inappropriate ADH Production. Making too much ADH, well, if you think about what is the job of ADH, it causes you to reabsorb more water. If you have too much ADH, then you're going to reabsorb too much water. If you reabsorb too much water, what happens? Your blood volume goes up, your blood pressure goes up, so you end up having all of the problems that come with hypertension, high blood pressure. On the other hand, hyposecretion of ADH causes diabetes insipidus. Now, we've mentioned this back in the kidney chapter, but we're going to go a little bit deeper now. Diabetes insipidus contains the symptoms polyuria, peeing a lot, that's what that means. Poly, much, or many, urea, urine. So you're urinating a lot. And remember, that's going to be the symptom for all diabetes. We're going to see in this chapter, there are many types of diabetes. Diabetes insipidus, polyuria, low blood pressure because you're losing water. Urine has a very low specific gravity because it's almost entirely water. But diabetes insipidus, there are two types. First, neurogenic, and second, nephrogenic. So if we look at diabetes insipidus, it's related to ADH. Maybe your body isn't producing or releasing ADH. That's either a problem with the hypothalamus or the posterior pituitary gland. That's neurogenic. Or on the other hand, maybe your body is making ADH just fine, but your kidneys aren't responding to it. That's a problem with the kidneys. That's called nephrogenic. So in neurogenic, ADH is either not being produced or it's not being secreted. Typically, this comes from something like a blow to the head or a brain tumor near the hypothalamus or the posterior pituitary. Something is actually interfering with these neurons and their ability to make or release ADH. In the case of nephrogenic diabetes insipidus, ADH is produced and secreted just fine, but your kidneys aren't responsive to it. Typically, this is genetic. So the body doesn't have the gene to make aquaporin type 2. So you can stimulate these cells with ADH, but they can't respond to it because the gene for aquaporin is defective. How would you actually be able to diagnose someone with neurogenic versus nephrogenic? What is something that you could test to see, do they have neurogenic or nephrogenic diabetes insipidus? Think about that. Use it as kind of a thinking cap question and see if there's something quick that you could do. Well, if you drew someone's blood, you could look to see their ADH levels. In neurogenic diabetes insipidus, they would have very low or no ADH. They're either not making it or they're not releasing it. In nephrogenic diabetes insipidus, they would actually have extremely elevated ADH because their body is trying to tell the kidneys, hey, we need to reabsorb more water. We keep getting dehydrated, so the ADH keeps being released. But the kidneys don't have the gene to respond, so they can't make aquaporin type 2. You can't reabsorb as much water. The body keeps releasing ADH from the posterior pituitary. It's just kind of an endless cycle. Next, let's look up here at the periventricular nucleus. This is where oxytocin is made. So oxytocin, its stimulus, or its stimuli, include cervical stretching, stretching of the cervix of the uterus, suckling when an infant goes to nurse, and intimate touch when you're either holding a baby or you are being intimate with your romantic partner. Its inhibition is just stopping those stimuli. So the cervix stops being stretched, or the baby stops suckling, or you stop being intimate. The functions of oxytocin: uterine contractions, labor. Back in Bio 137, when you were talking about feedback loops, there were only one or two feedback loops that were positive that are typically used as examples. One of the positive feedback loops in the body is delivery of a baby. So the way that oxytocin works is when a baby is ready to be born, it pushes against the cervix of the uterus. The uterus cervix gets stretched. That stimulates the hypothalamus to release oxytocin from the posterior pituitary. Oxytocin levels rise. Oxytocin causes uterine contractions, which pushes the baby against the cervix more, stretching the cervix, which causes more oxytocin to be released, which causes more contractions, stretching the cervix more, causing more oxytocin to be released, and that keeps going until the baby is delivered, and then the cervix stops being stretched. Milk ejection. So this is not milk production, but milk ejection from the nipple. This is when a baby is suckling. And the last one, love. And this one's always kind of interesting because everything that we associate as the feelings of love when we're, when we first met that person and, we've either been kind of cuddling with them or we've been intimate with them for the first time, or similarly, if you're holding your newborn baby. Well, all of these things trigger all of these warm feelings, but all of those warm feelings are the result of oxytocin binding to just the right spots in your brain. Love comes down to oxytocin, which is why if you think about the first time you were intimate with someone, chances are you could not wait to be intimate with them again. And for quite a while after, they are all you can think of, and you've got those butterflies in your stomach. Well, that's oxytocin. All right. So next time you think you're going to tell your partner that you love them and you want to tell them how special you are, really you can shorten it all and tell them, oh man, you give me such an oxytocin release, and leave it at that and see what kind of weird looks they give you. All right. So that's where we'll stop today. Next lecture is going to be focused on the other part, the anterior pituitary gland, and its relationship to the hypothalamus. So, like I said, there's going to be quite a few shorter lectures in this chapter, and that's because I know it's going to be rough sitting here basically listening to me read to you this whole time. All right, take care, and I will talk to you soon.